Bacterial cells can become infected by small viruses called bacteriophages. These bacteriophages insert their DNA into the bacterial cell. It is then transcribed, which will lead to the production of multiple more bacteriophages. As a means of adaptive immunity to infection via bacteriophages, the bacterial cells behold what's called the CRISPR locus. The CRISPR locus is a series of DNA which consists of repeated DNA sequences interspaced by spacers termed protospacers. Once bacteriophage DNA enters the bacterial cell, it will become degraded, and small pieces of the degraded DNA will be inserted into the protospacers. Now, CRISPR stands for Clustered Regulatory Interspaced Palindromic Repeats. The direct repeats of the CRISPR locus are all followed by protospacers. These protospacers have acquired bacteriophage DNA. The CRISPR locus, once transcribed, will be cut into various segments, each segment consisting of the direct repeat and the protospacer to which it follows. Each of the transcribed CRISPR repeats followed by the protospacer will associate with a Cas9 enzyme. If a bacteriophage is to infect the bacterial cell, some of its DNA may be complementary to the sequence within a protospacer. If this is the case, the protospacer will direct the CRISPR RNA and the Cas9 enzyme to this sequence of DNA within the bacteriophage. Adjacent to the sequence of bacteriophage DNA, there must exist a protospacer adjacent motif, that is, a short sequence of DNA bases which is recognized by the Cas9 enzyme. The Cas9 enzyme then induces a double-strand break within the bacteriophage DNA. Scientists can use CRISPR to target DNA of other organisms. By modification of the protospacer to any DNA sequence of interest, they can direct the CRISPR machinery to any genes they wish to be modified.